and welcome to everyone who already joined today's masterclass, and of course, hello to everyone who's going to watch the recording afterwards. My name is Marina Löwe. I'm a part of the service team here at Cube Biotech, responsible for the project management of our service projects. And today I'm pleased to moderate today's new episode of Scientific Masterclass series hosted here by Cube Biotech. So Cube Biotech's Scientific Masterclass series enables researchers from around the world to present their latest results and findings to a worldwide audience. This series provides a unique platform for scientists to share their research, their work, connect with others in the field, and receive feedback from the experts. Before we start and I introduce our today's speakers, I'm going to explain you how it's going to work. So we're going to hear the talk from our speaker today, and of course, we're going to have a question and answer session afterwards. And of course, you can write your question here in the lower right corner of your screen, and we're going to highlight your question afterwards on the screen and address it to the speaker. In case that we have too many questions and maybe we run out of time and cannot cover all of the questions, we're going to forward the questions to the speaker afterwards as well. So let's start. So our today's speaker is Elissa Moller from the University of Maryland in the USA. And Elissa is a PhD candidate in biophysics field, and she's studying the structure and function of mechanosensitive channels. She's co-supervised by Dr. Doreen Matthies in the Unit of Structural Biology at the National Institute of Child Health and Human Development, as well as by Dr. Sergei Sukharev in the Institute for Physical Science and Technology at the University of Maryland. And in today's talks, Elissa is gonna tell us about her insights into the structure of the mechanosensitive channel MscS in a native lipid nanodisc, and gonna show us her insights into understanding of the intramolecular forces and protein-lipid interactions that stabilize various protein conformation in the native lipid environment. So with this, I wanna ask Elissa to turn on her cam and the microphone. Hello, Elissa. Very nice to have you today. So,
thank you so much.
Thank you. I'm really looking forward to your talk. So stage is yours, yeah? You can start.
So let me get my presentation. Can you guys see my screen? Yeah. Okay. Hi, everyone. Thanks again for that introduction. My name is Elissa Moller. I'm a biophysics PhD candidate at University of Maryland, co-advised at NIH. And today I'm really excited to share my work with you guys on the structure of the mechanosensitive channel MscS in native lipid nanodiscs using the glycodimma polymer. So mechanosensation, which is the ability to sense environmental mechanical stimuli, is conserved across all organisms. And mechanosensitive channels are responsible for several essential functions, including hearing, touch, blood pressure sensing, cell volume regulation, and stretch-induced muscle and bone development. My focus is on MscS, which is a model system for mechanosensitive channel gating. It's a bacterial channel that opens in response to water influx during external osmolarity changes. And then the efflux through these channels reduces the osmotic gradient and prevents cell lysis. These channels improve osmotic fitness, enabling pathogens to colonize the osmotically challenging host environment and survive transmission through fresh water, causing outbreaks. They've also been implicated in antibiotic resistance, both decreasing the efficiency of membrane-targeting antimicrobials and increasing the sensitivity of other antibiotic classes, so they're attractive drug targets as well, due to their relevance to human health and disease and their extension to general mechanosensitive channel gating. Both structural and functional characterization of these channels is essential. I'm sure this audience is quite familiar with standard membrane protein purification protocols, but here's an overview of the different ways to prepare a membrane protein for structural determination. What I want you to take away from this is that in all cases shown here, lipid removal is the first step. This is most commonly done with detergents, but they're a poor mimic for the lipid bilayer environment and can cause structural changes or even protein denaturation. However, if your protein can withstand this initial detergent solubilization, you can reintroduce lipids in multiple ways. Most commonly, you see mixed micelles, liposomes, or synthetic nanodiscs. But that lipid removal step was still prerequisite for all these preparations. One unique feature of MscS is that being a mechanosensitive channel, it's highly sensitive to its lipid environment. There have been many structures of MscS since the original crystal. And the really remarkable thing is the dramatic transmembrane helical rearrangement when comparing the structure solved in pure detergent to anything that introduces a full-length lipid. So this structural change occurs even when you have DDM solubilized protein and you simply add a few lipids back into the solution to form a mixed micelle. And then a similar pair of structures has also been seen in synthetic nanodiscs based on the length of the lipid acyl chains. So the short chain lipid nanodiscs resemble the detergent solubilized structure, whereas the full-length lipid nanodiscs resemble the mixed micelle structure. And there's many, many examples of these two structural categories, which overlay almost exactly with the first two crystals, the original non-conductive structure, and then the crystal of the A106V mutation, which is conductive. But the goal of any of these structural studies is always linking it back to function. So we want to assign these structures to a place within the MscS gating cycle. MscS has three functional states that I'll be calling open, closed, and inactivated. These latter two are non-conductive. The inactivated state is non-conductive and tension insensitive, whereas the closed state is ready to open with increased membrane tension. The role of this inactivated state is to preserve osmolytes in the event of a non-lytic shock and allow for tension reduction below the opening threshold to fully reseal the membrane. A really central question in the field of mechanosensitive channels has been assigning the non-conductive structure of MscS to either the closed state or the inactivated state. And now the way we understand this gating cycle is by looking at the transitions using bacterial patch clamp electrophysiology. And this is done in native E. coli membranes. And the way it works is you take your bacteria, you treat them with a drug that keeps them from dividing. So they form these nice long snakes here. And then you can carefully lyse just the outer membrane so that your inner membrane, which is where these channels are, will collapse into a giant sphere that you can then pick up with a glass pipette, excise the remaining spheroplast, and then record the channel activity from this little lipid patch at the tip of the pipette. So you can do, you apply voltage and pressure, and what you're seeing here is each of these little blips is one single channel opening in response to that pressure application at both positive voltage and at negative voltage. Down here, you can see protocols that are examining population level opening, closing, inactivation, and recovery. And from this, we can get all the gating parameters that I'm showing here on the left. Since this channel is so sensitive to its lipid environment, we thought it would be really valuable to solve the structure in a native nanodisc, which uses polymers to extract membrane proteins directly out of the lipid bilayer without the need for detergent solubilization. So I used a polymer called glycodigma down here to solve the structure of MscS in a native nanodisc. I also tried two other polymers as well, SMALT 200 and acyclopol, which is now commercially available as ultrasolute amphipol 18. This is the full detailed protocol. I recombinantly overexpressed MscS with a 6-his tag. I used a French press to lyse the cells. I isolated the membranes, and then I did a second centrifugation step with a 3-step sucrose gradient to separate the inner membrane from the outer membrane. And the extraction efficiency of all of these polymers was much higher on this isolated inner membrane fraction when compared to the whole membrane fraction. And this is presumably due to the lipid composition. And then once I isolated those inner membranes, I did the extraction for 2 hours at room temperature. And then I took those extracted proteins and purified them for my protein of interest using cobalt affinity chromatography. I dialyzed away the imidazole as the final step, and then put it directly onto an EM grid. The sample looked good on both the STS and the Western, showing that it's relatively pure and confirming the identity with the His tag. But the Blue Native page was pretty inconclusive, with the majority of the signal remaining in the loading pocket and not running down into the gel. Despite that, the sample looked quite promising, albeit very heterogeneous, in negative staining EM. You can see both single particles as well as large membrane patches, which may be the reason that it didn't run well through the gel and stayed up in this pocket. Despite the heterogeneity, the negative staining data collection clearly shows MscS 2D class averages, both from the single particles as well as the larger membrane patches where the lipids can actually be seen extending all the way to the edge of the box. I also did some negative staining on the polymer alone as a comparison. The grid preparation was done in a relatively standard fashion, but I'll go through and highlight the unique aspects that optimize the grids for data collection. I used a 2 nanometer carbon support film grid to allow for lower protein concentrations and ensure that the particle ended up in the holes. And then to keep that carbon film intact, I could only glow discharge these grids for about 12 seconds. I used 0.3 mg per mil of the sample, and then I did a double sample application where I applied 3 microliters, blotted it off by hand as you would if you were preparing negative staining grids, and then I reapplied an additional 3 microliters of the sample before blotting it with the Leica. Once the grids were optimized, the data collection and processing was actually relatively straightforward, and it resulted in a 3 angstrom map. As I said before, I also tried 2 other polymers, SMALT 200 and ultrasolute amphipol 18, but these didn't result in structures, despite the fact that the grids appeared much more homogeneous. But you can already see at the 2D class average level that the transmembrane domain is fuzzy. And then in 3D, there's no density information above that cytoplasmic, soluble cytoplasmic domain. It's possible that this glycodimma polymer, with its variable size, acts a little less aggressively within the membrane and better preserves the transmembrane domain of this particular mechanosensitive channel. You can also see these variable membrane patches in the 2D class averages from Cryo-EM. So you can see that in some cases, the membrane boundary extends all the way to the edge of the box. And these classes look very, very different than what was actually used for the final reconstruction in the native nanodisc on the right. These liposome-like classes have unique top views as well. They're a little bit tighter and kind of sharply star-shaped without a clear opening for the pore. This may be a more compact structure, and or the density of the liposome on the other side might be seen through the pore of the protein. Unfortunately, I couldn't get a good 3D structure from these classes as I'm lacking the information in the transmembrane domain again. I'd need both a higher particle number and also to carefully subtract the membrane density from each of these particles to improve the signal to noise. But regardless, it's really, really cool to see that this polymer allows for direct membrane extraction and is gentle enough to maintain so much of the surrounding environment. So here again, I'm showing the structure that we could solve from those truly isolated particles, right? So the smaller nanodiscs that were just around my protein of interest. And in the cytoplasmic domain, the resolution reached about 2.5 angstroms, which was really exciting. And here you can see the model inside the mesh density, and then this is the map colored by chain. So the next thing that we wanted to do was know how this structure compared to the previously published structural categories. So this is the newly solved structure in glycodimma in comparison to the synthetic nanodisc structure with full-length lipids. This was the most similar structure overall with an RMSD of 0.9 angstroms. And it was also very similar to the mixed micelle structure as well. It's very different, however, from both the short-chain lipid nanodisc structure and the pure detergent structure. This latter one actually has an RMSD of almost 10 angstroms. What was really exciting is that there were additional densities in the native nanodisc structure that were not accounted for by the model protein, and some of them could easily be modeled in as lipids. This is where the lipids sit in respect to the protein. The lipids are shown in yellow, with lipid 1 up near the cytoplasmic leaflet, up near the periplasmic side of the membrane, and then lipids 2, 3, and 4 near the cytoplasmic leaflet. But the interesting thing about these lower lipids is that they're in distinctly non-bilayer positions. So the tails are actually facing inward toward the core of the protein, and then the head groups are facing out. And the general position of these lipids is similar to what we see in previous structures that reported densities for detergent molecules. These are shown on the right in gray. So lipid 1 has been previously reported in multiple synthetic nanodisc structures, and lipid 4 has been occasionally seen as well. Only one structure reported a lipid nearby where we see lipid 3, but it didn't overlay very well with our density. And then lipid 2 is actually newly reported in these structures. We went ahead and modeled these lipids as POPE based on mass spec lipidomics, comparing the isolated inner membranes to the lipids that co-purified with MscS in these native nanodiscs. And there was no significant lipid enrichment seen in that mass spec. So we just went with POPE as it's the most common lipid in the E. coli inner membrane. And so yeah, here I'm showing each of those POPE molecules inside their density, and you can see that the head groups of these lipids are coordinated by a handful of charge residues. So we've got R88, D67, R59, and K60, among others. We did some MD simulations as well that I don't have time to show you today. And we see that these lipids form a dynamic H-bond network, making these interactions very, very stable due to the multitude of configurations. And so what we wanted to do then is understand the role of these lipids in gating. So we went ahead and introduced mutations to these charged lipid coordinating residues. I won't go into all the details of these mutations, but I'll just highlight one triple mutant of the residues coordinating lipids 2, 3, and 4 that when we removed those charges had a dramatic non-inactivating phenotype, suggesting that these lipids down here are probably rearranged during this transition. It's important to note that the timescale of the inactivation recovery process would allow for lipid rearrangement, whereas the timescale of opening and closing would not. Since destabilizing these lipids decreases the likelihood of inactivation, this lipid-filled structure that we see probably represents the inactivated state of this channel rather than the closed state. Mechanistically, this is kind of logical, since the tension-transmitting helices, TM1 and TM2, are decoupled from the pore lining helix that contains the gate. This is TM3A. And then that gap is filled in by these 3 lower lipids. And then this also answers the question of which state the solved non-conductive structures belong to, since they have a very similar architecture to our native nanodisc structure. So in conclusion, we solved the structure of MscS in native lipid nanodiscs to 3 angstroms, extracted with the glycodipma polymer, which eliminates the detergent solubilization and lipid removal step that's common to all the previous structures of MscS. And this is a novel methodology for cryo-EM studies with really broad applications for the field. So it was really exciting that this worked. We observed densities of endogenous phospholipids that uncouple the gate from the peripheral tension sensing helices and interrupt the tension transmission route, representing a novel lipid-mediated mechanism of channel inactivation. And finally, I'd just like to leave you with some pros and cons from my limited experience with these polymers. comparing polymers and detergents. The biggest issue that I had with the polymers that I tried, at least, was the extraction efficiency was kind of low, meaning that a lot of my sample was lost in the insoluble fraction. I overcame this in two ways. One, just kind of brute force increasing the volume of my starting culture and having a larger amount of protein going into the experiment. And then, like I said earlier, extracting at room temperature from the isolated inner membranes. Both of those things help significantly. The resulting grids that I got were very heterogeneous, but don't let that dissuade you. With a larger data set and very careful processing, the structure could still be solved in my case. And then further separation by size, either with size exclusion chromatography or gradient centrifugation, would really improve the sample. And it's really promising that all of these lipids remain so stably associated with the protein. This let us resolve these native lipids rather than synthetic lipids. And also these really large membrane patches are very interesting. They might have less disrupted membrane bilayers and would be really viable for structural characterization in the future. For MscS specifically, the use of this polymer increased the predictive power of our Cryo-EM structure and allowed us to associate the solved conformation with the inactivated state. So yeah, I'd like to thank my advisors, Dr. Sergei Sukharev and Dr. Doreen Matthies, and all the lab members, especially Dr. Madeline Britt and Dr. Fei Zhao. NICE did the Cryo-EM data collection over at NIH, and then University of Maryland, Baltimore did the mass spec. My funding sources are NSF and NICHD, and then NIH BioWilf provided all the computational resources. And with that, I'd be happy to take any questions.
Okay, thank you very much. This is exciting talk, yeah. You know, this is also one of the things that I addressed partially during my PhD and what we're trying to address, you know, to people right now, yeah. When you're working with membrane proteins, lipids do matter in the end. So you cannot look at them, you know, separately. Yeah? So this is the one organism, so to say, yeah? So that you're working with. Yeah? So and it's pretty cool to see, yeah, so that he was able really to go forward, yeah, so with this nanodiscs, yeah, so and see those amazing structures. I will take a look on the questions that we have right now. Can we highlight it like this? Okay, we have a question. So how did you arrive at the glycodigma as the ideal polymer for your extraction and purification, and how much polymer do you use for a typical purification?
So I wish I had a logical way to get to glycodigma, but the honest answer is I actually received a free sample from Cube at a conference and gave it a try. I tried the other two polymers previously because we already had SMALP in the lab, and then the people who were developing cyclopoles gave us a sample before it was commercially available. So it was just 3 random choices. And yeah, so I don't have a super concrete way to do it. I think it would be similar to a detergent screening where you just try a bunch of polymers and see what ends up working the best. And then for the amount that I did, I always tried to use the lowest recommended polymer concentration. So in the case of glycodigma, I did 0.5%.
Yeah, perfect. This is exactly what also the thing that we advise always. Yeah, so with the screening approach. Yeah, so because this is like a detergent screen. Yeah, so you can guess which one maybe can work. Yeah, so the best for your application in the end and for your target. But in the end, it's so target specific, you know, and the polymer specific that we really advise also to make a bit larger. So it maybe should not be, you know, like super big screen. Yeah, so but you. May test different substances, yes, and see which perform best, because you also mentioned that the yield was not very fine, yeah, so at the beginning, so you needed to tune it, yeah, so with some polymers, yeah, so you see this in the screening, yeah, so how it behaves, yeah, so and then can judge, yeah, so basically which one then is the best. Thank you.
Yeah, and there's beautiful structures in SMALP 200 and cyclopoles that are published, right? So it just didn't happen to work for my protein.
May happen, of course. Thank you. So let's go to the next question. Thanks. You said at some point you had a short-chain lipids in nanodiscs with your structure. Did you first reconstitute into liposomes and then re-extract by glucodigma?
So the short-chain lipid nanodisc structure that I was discussing was actually previously published by a different group. And those are in synthetic nanodiscs that are formed with MSPs. So that's the typical setup where you do the detergent extraction and then you reconstitute. And they purposefully use short-chain lipid nanodiscs in order to see if they could force an open state of this channel. I used full-length native E. coli lipids because I extracted directly from the native membrane. So I did not do any detergent solubilization, no liposome reconstitution, just direct extraction from the native inner membrane. I did isolate the inner membrane, but that was the only step I did before adding the glycodigma.
Yeah, I think that could make sense. Yeah, so to use also some different membrane thicknesses. Yeah, so to induce this changes now. So yeah. So they probably would react, yeah. So especially this type of the proteins would react on this change.
Channel is very sensitive.
Yeah.
Yep.
Okay, let's go to the next one. Do you think glycodigma has affected the functional state from active to inactive state?
That's a really good question. So this channel, we are thinking that the default low energy state of this channel is actually the inactivated state. And we might need a little bit more lateral pressure from a full bilayer, or even some of the gradients that are held across the bilayer might be important to getting it to the closed state, which is then ready to open. So I would say that it's possible that the glycodigma did shift the distribution more toward inactivated, but it's also possible that that's just kind of the default resting state of the channel. That's still functional characterization that we need to do further.
Oh, interesting. Maybe I can jump in also with a question. Yes, so because you tried 3 different polymers. So have you seen the differences basically in the sizes of the discs? Yeah. So yes. So maybe you can give a couple of insights. Yeah. So what happened with the different polymers there?
Yeah. So for my limited experience, right? So for the SMALT 200 and the cyclopoles, the amphipole 18. So they are both much tighter nanodiscs, much smaller, much closer associated with my protein. Whereas with glycodigma, we see a range of sizes. So there's very small ones that are tightly associated, and then much, much bigger membrane patches as well. So the grids looked better from like somebody who's used to looking at detergent solubilized membrane proteins. The grids for the ultrasolute amphipole 18 and the SMALT 200 looked much better by eye. But once I collected the data, I think those polymers seem to have disrupted the transmembrane domain somehow, maybe by associating too tightly, trying to form too small a nanodisc. I'm not really sure. But glycodigma had those giant membrane patches as well as reasonable sized nanodiscs where I could still resolve the transmembrane domain.
Yeah, for sure. And they probably gave more, flexibility to the protein. So because they're kind of a bit larger. Yeah, so cool. Thank you. So, well, the next question. What size discs were generated with glycodigma? Would this have an effect on the structure? I think we just
yeah, sorry, we jumped ahead of you there. But yeah, the discs were super variable, from very, very small around the protein to absolutely huge membrane patches. So yeah, and that definitely did affect the structure.
Perfect. Thank you very much. So let's go to another one. Okay, it seems that the side that the lipid chains plays a role in the MscS structure. Any idea how tension packing in a nanodisc plays a role in the structural changes of this protein?
Yeah, so that's kind of that's basically what I'm working on for my PhD, right? So the lateral pressure of a full bilayer is very, very different than the lateral packing that you are going to see in one of these nanodiscs. And we were hoping that the major disrupting force would be the detergent, and we might see a new structure in these native nanodiscs. It turned out that we saw a very similar structure, but we could still get a lot of insight into the function of those lipids, which was awesome. But it does seem that these native lipid nanodiscs are still not packed exactly the same as you would see in a full membrane. So there's definitely, the lipid chains are very, very important for the MscS structure. It's very, very sensitive. It's a low tension threshold channel. So it's definitely has an impact on the structure. I don't know if that answers your question. If I can clarify more, I'm happy to.
Yeah, I mean, we can. Yeah, so this, yeah. Address the question, please, in the box. Yeah, so then we may discuss further. Yes. Okay.
Perfect.
Okay, next question. Was the extraction efficiency for amphipol 18 and other polymer higher than glycodigma, or basically we may compare, yeah, so the extraction properties? Yeah.
Yeah, so the extraction efficiency might have been a little bit higher for the other polymers. Overall, they're all much lower than detergent, which is to be expected. But the extraction efficiency wasn't actually the deciding factor on what gave me the best structure. Because even though the extraction efficiency was low for glycodigma, I still was able to get the structure out at the end. So it's really, I think unfortunately, you can't just look at the extraction, because amphipol 18, for instance, extracted much, much better. And I was thinking that that was definitely going to do it. But then the resulting structure was not, you couldn't see the transmembrane domain, but probably because it extracted a little bit too well.
Yeah, that's also funny, no? So that the best yield is not always the best quality, yeah, so of the sample for the Cryo-EM, yeah. So that's also interesting, yeah. Sure. Okay, let's go to the next question. Do you observe asymmetry between subunits during different structures during activation or inactivation?
So I don't know that I have a good answer for that, because I don't know that I can say I can directly observe these structural changes. What I'm looking at, right, is an average of a static state that is actually a distribution, right? So it's a little hard to answer that directly, but we don't think there's asymmetry in the opening transition of this channel. From the patch clamp and some MD simulations, we think that it's just based on the lateral pressure of the membrane. So as the membrane stretches, these channels open relatively uniformly, kind of like an iris on a camera. So where all of the edges are expanding outward at the same time. So we don't see specific asymmetry for this channel.
Thank you very much. Let's go for another question. Or do we don't have questions anymore, I think. So guys, if you have some question more, yeah, so please type them, yeah, so maybe I will also ask some, yeah, so if you have some questions, you know, after the talk, after 1 hour or something, you think, oh, I should have asked this one and this one, you can always write an email to us, and we're going to forward the question to Elissa. Maybe just just in general, yeah. So for a question, so you probably worked a lot with detergents previously, yes, and then tried the polymers. So what was your insight? Yeah. So have you needed to, or did you needed to, you know, adjust your protocols very differently, or just tell about your experience in working on polymers in comparison to the previous workflows that you did?
Yeah, so I was lucky going into this that the detergent extraction for MscS had been pretty well established. So I didn't have to do too much screening going into the detergent prep. Initially, of course, people had to do a lot of work to optimize the detergent conditions for this guy. But for the polymers, it was really a lot of trial and error, a lot of small scale purifications. I was focusing a lot at the beginning on extraction efficiency because that was where I was losing the most sample. And eventually I just kind of was like, okay, as long as some is getting extracted, I can scale up my purification to overcome that problem. And then isolating the inner membranes helped a lot. I think these polymers seem to be a little bit happier with E. coli inner membrane versus the whole membrane fraction that also has the outer membrane lipids, so no LPS and stuff like that seem to help it a little bit. And then the room temperature extraction, which I would never try with detergent, right, was really, really huge for getting this these polymers to work at all. I think maybe it's because of the packing in the 4 degrees, right, of these lipids, the phase of those lipid tails. So it definitely, room temperature, I was very scared to try. And I only did it for 2 hours. I know people have done it at higher temperatures and for longer times successfully. I was worried about damaging my protein, and I was like, 2 hours gives me enough, so I'm just going to call it there. But the room temperature was a huge, huge help.
Yeah, this is the crazy story as a protein chemist. Yeah, so then you always work in ice. Yeah, so and then you drag your sample on the table, you know, and work like this. Yeah, so that's a bit, you need to change your mind a bit with the polymers. Yeah, so because exactly what you told. Yeah, so the lipids, they do matter. Yeah, so and the lipids, they physical chemical properties at 4 degrees and on body temperature or the room temperature, they are completely different. Yeah, so for sure. Yeah, so and then of course, yeah, I mean, for those who is watching us, yeah, so and struggles with the solubilization and the purification steps, yeah, so and having low yield, so we always advise to make a screening, yeah, so to try different polymer concentrations, to try different polymers, yeah, so and maybe also the preparation is important. So what you sell, yeah, I told, yeah, so that from the inner membrane you get better results, so to say, yeah, so and that's always worth to try it, yeah, so in the end, yeah, so and optimize your protocol in this way. So because we have on detergents already so many years of experience, you know how to work with this, yeah, so with polymers, of course, yeah, so that's maybe not everyone and not every membrane protein expert maybe worked already with this, so we need to, you know, to get into, yeah, so and try all things, yeah, so and establish really this workflows, yeah, so and make great results, because I think that's great, yeah, so what you just showed, yeah, so and my heart, you know, it's always, you know, flattering when I see this beautiful lipid structures, yeah, so together with the protein. So, we have another question. Did you also try extraction at 37 degrees, especially since E. coli prefers 37 degrees?
I did not try 37. I think that definitely is worth trying. I was a little hesitant because I was very worried about the stability of my extracted membrane protein, but based on how well it did at room temperature, I think 37 would absolutely be worth trying. And I've seen it in the literature. People do have the heated rotisseries for the extraction. So it definitely is worth trying. I didn't try it, but I think it would be fine.
Yeah. For sure. So since we have no questions left, I want to thank you, Elissa, one more time for coming today. Yeah, so thank you for the exciting talk. I wish you all the best, yeah, so for the future research, yeah, so and I hope we'll see us in the person in some conference maybe in the future. Yeah, and yeah, thank you very much.